Hello, thank you for interested in my poster. And the study I'm going to present today is nascent extracellular glycoprotein profiling during critical lung stages, a nascent glycoprotein atlas for studying lung rejuvenation. So, as you know, the lung is a critical organ for gas exchange, and aging is an inevitable biological process that leads to lung functional decline due to elevated inflammation, fibrosis, and loss of regeneration ability. Therefore, understanding the mechanism of lung development and regeneration is critical for studying lung rejuvenation. The human lung development involves multiple stages, while the in vitro differentiation of iPSC successfully recapitulated the prior stages. Um, our understanding in alveolar epithelial cell maturation stage, which corresponds to alveologenesis in development, remains limited. And therefore, deeper understanding in protein dynamics during alveologenesis will provide us critical information to study lung rejuvenation. So the discovery of central dogma has enabled us multiple levels to understand protein expression dynamics. Genomics helps us understand what can be expressed, but not where, when, or how much. Transcriptomics tells us the instantaneous levels of mRNA, but sometimes the correlation between mRNA and protein levels can be elusive. Conventional proteomics characterizes the amino acid sequences with mass spectrometry. However, due to the unamplifiable nature of amino acid sequences, the abundant proportion-based mass spectrometry yields a high background signal. And meanwhile, most structural or supportive proteins that do not change are normally of high abundance, while the signaling proteins that undergo dynamic changes are often of low abundance. So therefore, selectively labeling and characterizing of newly synthesized proteins, also termed nascent proteins, bring huge benefit to understanding of protein dynamics. So here we introduced azidogalactosamine and azabarine monosaccharides to mice or rats, which can be incorporated onto glycoproteins via new protein synthesis for metabolic labeling. We aim to apply this method to three different lung stages: lung development, regeneration, and aging, to selectively study nascent proteins. And here, our immunofluorescence stainings shows robust and specific AZ taggings on lungs in adult mice and neonatal rats. Many previous studies of lung rejuvenation are focusing on the cell behavior. However, emerging evidences are highlighting the critical roles of extracellular matrix, the ECM plays for guiding development. Here, we use the stepwise extraction method to remove the cytoplasmic contents and selectively study proteins that connects to the ECM. And our proteomics suggests the most abundant proteins we extracted are membrane and ECM proteins here. We then chemoselectively purified the AZ-tagged extracellular proteins using affinity purification. So basically, our AZ-tagged proteins are destillbutylated, pulled down by streptavidin beads, and then eluted with belting with a competitive manner. And we observed specific protein signals from AZ label groups compared to vehicle control. We then applied this technology to adult and aged mouse lungs and identified 89 proteins with elevated synthesis levels and 195 with declined synthesis levels in aged lungs compared to young adults. Among these identifications, we observed many proteins with similar patterns for aged lungs in other studies, such as laminins, warm-body benefactors, and tenacin C. We will continue to apply this technology to lung development and regeneration to identify critical markers for alveologenesis. So, in conclusion, we established three models to highlight distinct biological stages of lung development, regeneration, and aging. We chemoselectively characterized nascent extracellular glycoproteins in aged lungs as a proof of concept and identified a few interesting protein synthesis patterns. And eventually, by applying this technology to neonatal rats and adult rats after receiving pneumonectomy, we will generate a nascent extracellular proteome atlas, which will potentially help us identify novel biomarkers and targets to boost alveoli maturation and regeneration. So we foresee this proteome atlas to offer novel insight for the study of lung rejuvenation. Thank you for your attention, and I would like to thank all the lab members and collaborators with their extensive helps. Thank you.